Hello students, welcome to academia. Today I will discuss an important endocrine gland which is pancreas. Generally, pancreas is both exocrine and endocrine gland. The exocrine part secretes pancreatic fluid into the duodenum after a meal, and the endocrine part secretes various types of hormones. So, what are the types of pancreatic cells which can produce different hormones? Number 1, A cells. They produce glucagon. Then B cells which produce insulin. D cells which produce somatostatin, and F cells which produce pancreatic polypeptides. These endocrine cells of pancreas are situated in a specialized tissue, and the region is known as islets of Langerhans. It represents approximately 1 to 2% of the pancreas. The pancreatic hormones are responsible mainly for the storage of glucose in form of glycogen, and they enable the mobilization of energy reserves as a result of food deprivation, stress, and physical activity. They maintain the constant plasma glucose concentration also, as well as stimulate growth. So, today we see the important hormone of the pancreas, that is insulin. So let's start. Now, what is the structure of insulin? The human insulin contains 51 amino acids arranged in 2 polypeptide chains, chain A having 21 amino acid and chain B having 30 amino acid. 2 interchain disulfide bridge is there, and interchain disulfide link in chain A. So, in the diagram, can easily see the arrangement of amino acids in the insulin, and where we found the disulfide bridges also. The insulin is basically a protein, and we know that the proteins are synthesized within our cell. The gene for protein synthesis is located in the chromosome 11, and they are produced by the beta cell of the pancreatic islets. The synthesis involved 2 precursors, that is pre-proinsulin 108 amino acid and proinsulin, that is 86 number of amino acid. In the diagram, we can see that how the insulin is stimulated to synthesis. The pre-proinsulin, from this there is proinsulin and then the insulin. Insulin secretion has a pulsatile character, and its main stimulus is an increased blood glucose level. When after eating something, after absorption, there is increased level of glucose in blood, then the insulin receptors are stimulated. Now, how it occurs? So, we can clearly see in the diagram the process of the stimulation. The increase in plasma blood glucose level, increase in the value of glucose in the B cell, then its oxidation increases and thus the amount of ATP in the cell. Then energy controlled K+ channels close, which is due to the depolarization, and the voltage controlled calcium channels open. Increased level of calcium cations in the cell cause insulin exocytosis and reopening of the K+ channels. Numerous other factors also stimulate the insulin secretion during the digestion process. So this is the way how the insulin is secreted after the stimulation. About the introduction of insulin. In this slide, we see the functions of the insulin and how it works. So, this is all about the introduction of insulin. Hope you can understand. If you have any query and question, you can ask during the class or comment in the comment box.